This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wonder how life survives in the most extreme environments? This article explores the fascinating world of symbiosis, where organisms team up for survival. We'll dive into a unique partnership between a ciliate and a bacterium, thriving in an oxygen-free lake. Using easy-to-understand language, we'll uncover how this bacterium acts as a tiny power plant, generating energy for its host through a process called denitrification. Prepare to be amazed by the ingenuity of life and the surprising ways organisms adapt to even the most challenging conditions. Chapter 1. A Symbiotic Pact: How a Bacterium Became a Ciliate's Power Plant. Such a place exists in the sunless depths of certain lakes, where the water stagnates and oxygen disappears. Yet, even in these seemingly barren conditions, life, tenacious and resourceful, finds a way. In the murky depths of Lake Zug, a picturesque Swiss lake nestled amongst the Alps, a peculiar ciliate, a single-celled organism propelled by hair like cilia, thrives. This isn't just any ciliate. It harbors a secret, a hidden partnership that fuels its existence in this oxygen-deprived world. Our story begins not with the ciliate itself, but with a team of scientists at the Max Planck Institute for Marine Microbiology. Initially, they were investigating the microorganisms involved in methane metabolism in Lake Zug's deep waters. The lake is meromictic, meaning its layers don't mix, creating a unique environment in the depths. Anoxic, rich in methane and nitrogen compounds like nitrate, and isolated from the oxygenated surface world. As part of their research, they were analyzing the lake's metagenome, essentially the collective genetic material of all the organisms present in a sample. When analyzing a metagenome, scientists don't examine individual organisms. They look at all the genes present in the sample, searching for specific gene sequences relevant to their research. This is often the only way to study microorganisms in environmental samples, as many cannot be cultured in a laboratory. While searching for methane-consuming bacteria with genes related to nitrogen conversion, one of the researchers, John Graff, stumbled upon an anomaly. He found an amazingly small gene sequence, far smaller than expected, yet it contained the complete blueprint for respiratory denitrification, a process of energy production using nitrate. This was a surprise, as denitrification is typically associated with bacteria, not eukaryotes. But the real puzzle was the location. The sequence came from a sample taken from the anoxic zone of the lake. The initial hypothesis was that the genes belonged to bacteria associated with insects, a known location for similar genes. But insects in the deep, oxygen-free waters of Lake Zug? It didn't add up. The scientists were baffled. How could a complete denitrification pathway exist in such an environment, and more importantly, in what organism? The research group was abuzz with speculation. Guessing games and betting ensued, each scientist proposing a different explanation for the mysterious gene sequence. One prevailing thought emerged. Could the genes belong to an unknown endosymbiont, a bacterium living inside another organism? Endosymbiosis, where one organism lives within the cells of another, is a well-established phenomenon in biology. The most famous example is the mitochondria, the powerhouses of our own cells, which are believed to have originated from ancient bacteria. However, studying endosymbiosis presents unique challenges, primarily because most symbiotic microorganisms cannot be grown in isolation. To test their endosymbiont hypothesis, the team embarked on several expeditions to Lake Zug. They collaborated with the Swiss Aquatic Research Institute (EAWAG), collecting water samples specifically from the anoxic depths, around 197 meters deep. Back in the lab, they meticulously used pipettes to isolate individual eukaryotes from the samples, hoping to find the organism harboring the mysterious genes. They then employed fluorescence in situ hybridization, FISH, a technique that uses fluorescently labeled probes to bind to specific DNA sequences, allowing them to visualize the presence and location of the denitrification genes. One of these expeditions, in the winter of 2020, proved particularly memorable. The team faced stormy weather, dense fog, and the added pressure of the emerging COVID-19 pandemic and the threat of a lockdown. Retrieving samples from the deep lake became a race against time and the elements. Despite the challenging conditions, they successfully collected samples and transported them back to Bremen. The painstaking work paid off. Using fish with a specifically designed probe, they finally visualized the denitrification genes. Every ciliate they examined from the 180-meter depth contained these bacterial symbionts. Further genetic analysis confirmed that the ciliates belonged to the class Plagiophylia, a group known for inhabiting anoxic environments. Inside these ciliates, living in a symbiotic partnership, was the bacterium responsible for the unusual gene sequence. They named it Candidatus azoomycus ciliaticola, a name signifying nitrogen friend dwelling within a ciliate. The discovery of azoomycus raised more intriguing questions. Lake Zug is a relatively young lake, formed only about 10,000 years ago after the last ice age. Yet, the symbiotic relationship between azoomycus and its ciliate host is estimated to be much older, perhaps 200 to 300 million years old. How did this ancient partnership end up in a geologically young lake? The answer, it turns out, might lie in the broader distribution of azoomycus. Since the initial discovery, similar gene sequences have been found in various aquatic environments around the globe, suggesting a far more widespread presence than initially suspected. This raises fundamental questions about the origin of this unique symbiosis, its evolutionary history, and the mechanisms that allowed it to disperse across continents and through vast stretches of time. The story of azoomycus had just begun, and it was already rewriting our understanding of life's adaptability and the surprising partnerships that make it possible. Chapter 2. In the realm of nitrate, life without oxygen. The ciliate, our single-celled protagonist, resides in a world vastly different from our own. Imagine descending into the southern basin of Lake Zug, a beautiful Swiss lake cradled by the Alps. As you dive deeper, the sunlight fades, and the familiar blue transforms into an inky blackness. Around 197 meters down, you enter a realm devoid of oxygen, a silent, watery world known as the anoxic zone. Here, sophisticated instruments like micro-optodes and Clark-type oxygen sensors, capable of detecting even trace amounts of oxygen, confirm its absence. Yet, it is precisely in this seemingly inhospitable environment that our ciliate thrives. This deep, oxygen-free zone is a consequence of the lake's stratification. Like layers of a cake, the water separates into distinct strata, with the denser, colder water settling at the bottom. This prevents mixing with the oxygen-rich surface waters, creating a stable, anoxic environment below. Furthermore, this isolated layer becomes enriched with nitrogen compounds, particularly nitrate, which seeps in from the surrounding environment. Scientists, eager to study this unique ecosystem, carefully collect water samples using a Niskin bottle, a specialized container designed to minimize oxygen contamination during retrieval, ensuring the integrity of their precious cargo from this alien world. Our ciliate, a tiny, ovoid creature, measures only about 25 micrometers long and 20 micrometers wide, smaller than the width of a human hair. Its surface is covered in hair-like cilia, beating rhythmically to propel it through the water. Under a binocular microscope, using a delicate glass capillary, scientists painstakingly isolate individual ciliates from the lake water for analysis. These ciliates belong to the class Plagiophylia, a group known for its members' preference for oxygen-poor or oxygen-free environments, both in freshwater and marine habitats. Many Plagiophylians form partnerships with methanogenic archaea, single-celled organisms that produce methane. However, our ciliate stands out, having forged a different alliance, a pact with a bacterium that breathes nitrate. This bacterial ally, nestled within the ciliate's cytoplasm, has been visualized using a technique called fluorescence in situ hybridization, or FISH, which illuminates the bacteria within their host like tiny, glowing beacons. The bacterium, known as Candidatus azoomycus ciliaticola, belongs to a group called gamma proteobacteria. Azoomycus, meaning nitrogen friend, is more than just a friend. It's an essential partner, the first obligate endosymbiont ever observed with the complete machinery for denitrification. This remarkable bacterium possesses a streamlined genome, a mere 0.29 megabases in size, tightly packed with protein-coding genes. This compact genome reflects its specialization, energy production. It's a minimalist, highly efficient power plant, stripped down to the essentials for denitrification. Scientists, using techniques like clone fish and Sanger sequencing, have meticulously deciphered the secrets of this tiny genome, revealing its remarkable adaptations. The process of denitrification, carried out by Azomachus, is a multi-step chemical transformation where nitrate is converted into nitrogen gas, releasing energy in the process. This energy is captured in the form of ATP, the universal energy currency of cells. Azomachus doesn't hoard this energy, it shares it with its host. A specialized protein, the ATP-ADP translocase, acts as a shuttle, transporting ATP from the bacterium to the ciliate's cytoplasm, fueling the ciliate's activities. Interestingly, both the bacterium and the ciliate lack terminal oxidases, enzymes essential for using oxygen in respiration. This absence strongly suggests that they are obligately anaerobic, completely adapted to a life without oxygen. Within Azomachus, an intricate electron transport chain orchestrates the denitrification process. Electrons are passed along a series of protein complexes, ultimately leading to the conversion of nitrate to nitrogen gas. This flow of electrons generates a proton gradient, a difference in proton concentration across a membrane, which drives the synthesis of ATP. This ATP, then shuttled across to the ciliate by the ATP-ADP translocase, becomes the ciliate's primary energy source, allowing it to thrive in the oxygen-deprived depths. Further solidifying its commitment to an anaerobic lifestyle, the ciliate exhibits negative aerotaxis. This means it actively avoids oxygen, retreating from even the slightest whiff of this otherwise life-giving gas. This aversion to oxygen underscores the ciliate's complete dependence on its nitrate-respiring endosymbiont. This intricate partnership, a biological dance between a ciliate and its bacterial powerhouse, showcases the remarkable adaptability of life. In the absence of oxygen, where most eukaryotes falter, this ciliate has not only survived but thrived, thanks to the unexpected powerhouse within. It's a testament to the power of symbiosis, a reminder that even in the most extreme environments, life finds a way, forging unlikely alliances and rewriting the rules of survival. Chapter 3. Nitrate, the new oxygen? A bacterial powerhouse. This process, a multi-step biochemical transformation, allows Azomachus to extract energy from nitrate and share it with its host, rewriting the rules of respiration in this oxygen-deprived world. Let's delve into the intricate details of denitrification, contrasting it with the more familiar aerobic respiration that powers our own cells. In aerobic respiration, oxygen serves as the final electron acceptor in a chain of reactions that generate energy. Electrons, stripped from food molecules, are passed along a series of protein complexes, ultimately combining with oxygen to form water. This electron flow creates a proton gradient across the inner mitochondrial membrane, a difference in proton concentration that drives the synthesis of ATP, the cellular energy currency. Denitrification, however, takes a different route. Instead of oxygen, Azomachus uses nitrate as the terminal electron acceptor. This process unfolds in a series of precisely orchestrated steps, each catalyzed by a specific enzyme. First, nitrate reductase, an enzyme containing a molybdenum cofactor, converts nitrate, number 3, to nitrite, number 2. This initial step, occurring in the periplasm, the space between the inner and outer membranes of this gram-negative bacterium, sets the stage for the subsequent reactions. Next, nitrite reductase, another periplasmic enzyme, further reduces nitrite to nitric oxide, NO. Two forms of nitrite reductase exist, one containing copper, NERC, and the other containing cytochrome CD1, NERS. The specific type present in Azomachus would depend on the specific subgroup it belongs to. Nitric oxide, a toxic gas in our own bodies, is merely an intermediate in Azomachus's energy-generating scheme. The next stage involves nitric oxide reductase, which converts nitric oxide to nitrous oxide, N2O, also known as laughing gas. This enzyme, situated within the bacterial cell membrane, plays a crucial role in the denitrification pathway. Finally, nitrous oxide reductase, another membrane-bound enzyme, completes the transformation, converting nitrous oxide to nitrogen gas, N2. This final product, harmless and abundant in our atmosphere, represents the culmination of a Zooamicus's nitrate respiration. This stepwise reduction of nitrate, from NO3 to N2, releases energy that a Zooamicus captures and uses to generate ATP through its electron transport chain. This ATP is then transported across the bacterial membrane into the ciliate cytoplasm via a specialized protein called the ATP-ADP translocase. This translocase, similar to those found in mitochondria, allows a Zooamicus to effectively function as the ciliate's power plant. The energy yield of denitrification, while lower than aerobic respiration, is significantly higher than fermentation, the anaerobic process often used by organisms lacking mitochondria or similar organelles. Aerobic respiration, the most efficient of the three, generates approximately 32 ATP molecules per glucose molecule. Denitrification, while less efficient, still produces a respectable amount of ATP, ranging from 5 to 10 ATP molecules per nitrate molecule. Fermentation, on the other hand, yields only 2 ATP molecules per glucose molecule. This difference in energy output explains why the ciliate, powered by a Zooamicus's denitrification, thrives in its energy-limited environment, outcompeting organisms relying solely on fermentation. The genomic analysis of a Zooamicus reveals further adaptations to its endosymbiotic lifestyle. Its genome contains a high density of protein-coding genes, mostly dedicated to energy production. This compact genome, a testament to its specialization, lacks genes for many essential biosynthetic pathways, indicating its reliance on the host for building blocks like nucleotides, amino acids, and lipids. This dependence further solidifies the obligate nature of the symbiosis. Both a Zooamicus and its ciliate host lack terminal oxidases, the enzymes that would enable them to use oxygen for respiration. This absence, coupled with the ciliate's negative aerotaxis, its active avoidance of oxygen, strongly suggests an obligately anaerobic lifestyle. These organisms are not just tolerant of low oxygen conditions, they are completely adapted to, and dependent on, the absence of oxygen. Researchers, using a suite of sophisticated tools, have meticulously pieced together this story of symbiosis and survival. Geochemical profiling of Lake Zug, including dissolved oxygen measurements with microoptodes and CTD probes, confirmed the anoxic nature of the ciliate's habitat. These probes, lowered into the depths, provided precise measurements of oxygen concentration, temperature, pH, conductivity, and turbidity, painting a detailed picture of this unique environment. Nutrient measurements, using techniques like fluorometric quantification and segmented flow analysis, revealed the high concentrations of ammonium, NOx, nitrate and nitrite in the anoxic zone, the raw materials for a Zooamicus's denitrification. The bacterium and its host were identified and studied using techniques like FISH, single ciliate PCR, and Sanger sequencing. FISH, as mentioned earlier, visually pinpointed the bacteria within the ciliates using fluorescent probes. Single ciliate PCR, using primers designed specifically for a Zooamicus's 16 srRNA gene and the ciliate's 18 srRNA gene, allowed researchers to amplify and sequence the DNA of individual organisms, confirming their identities and phylogenetic affiliations. Sanger sequencing provided high-quality DNA sequence data, enabling detailed genomic analysis of a Zooamicus. Clone libraries, constructed by inserting a Zooamicus's 16 srRNA gene into plasmids and transforming them into E. coli, provided a stable source of DNA for further analysis. The story of a Zooamicus and its ciliate host is not just a tale of two organisms, it's a window into the vast, unexplored world of microbial partnerships. It highlights the remarkable adaptability of life, its ability to thrive even in the most extreme environments, forging unlikely alliances, and constantly innovating to overcome the challenges of survival. This unique partnership, a biological dance between a ciliate and its bacterial powerhouse, raises fundamental questions about the evolution of eukaryotes, the origin of mitochondria, and the potential for similar symbioses to exist in other anoxic environments around the globe. This discovery also hints at the broader role of denitrification in shaping ecosystems, its impact on nutrient cycles, and its potential contribution to the production of greenhouse gases like nitrous oxide. The subsequent chapters will delve deeper into the intricacies of this symbiotic relationship, exploring the evolutionary history of a Zooamicus, its interactions with its host, and its broader ecological significance. We'll uncover how this nitrogen friend has not only secured its own survival but has also enabled its ciliate host to flourish in a world without oxygen, a testament to the power of symbiosis and the remarkable ingenuity of life. Chapter 4. Echoes of the Past, a new endosymbiotic narrative. The discovery of the nitrate-breathing ciliate in the depths of Lake Zug has unveiled a remarkable example of evolutionary innovation, mirroring the ancient endosymbiotic event that gave rise to mitochondria. This modern-day symbiosis between the ciliate and its bacterial partner Candidatus Azoamicus ciliaticola offers a compelling glimpse into the dynamic interplay between prokaryotes and eukaryotes, and the remarkable adaptability of life in the face of environmental challenges. The origin of mitochondria, a pivotal event in the history of life, occurred over 1 billion years ago when an ancestral archaeon engulfed an alpha proteobacterium. This chance encounter, a biological merger of two distinct life forms, laid the foundation for the evolution of eukaryotic cells, the complex cells that make up all plants, animals, fungi, and protists. The engulfed bacterium, initially a free-living organism, gradually shed its independence, becoming integrated into the host cell and evolving into the mitochondrion, the cellular powerhouse responsible for aerobic respiration. The ciliate's partnership with Ca. Azoamicus ciliaticola represents a more recent echo of this ancient event, a separate evolutionary dance with striking parallels. Just as the ancestral alpha proteobacterium became an integral part of the eukaryotic cell, Azoamicus has become indispensable to the ciliate's survival in the anoxic depths of Lake Zug. A key parallel between the two endosymbiotic events lies in genome reduction. This compact genome, densely packed with protein-coding genes, is significantly smaller than those of free-living bacteria like Legionella clemsonensis, 3.27 megabits, and even smaller than other endosymbionts like Caeotibacter tiniospiralis, a bacterium found in other ciliates. This reduction in genome size is a hallmark of endosymbiosis, as the symbiont, residing within the protective environment of the host cell, can afford to lose genes that are no longer essential for its survival. This process of genome reduction is ongoing, evidenced by the presence of pseudogenes in Azoamicus's genome, remnants of genes that are no longer functional. The mitochondrion, the product of an ancient endosymbiotic event, has undergone even more drastic genome reduction, retaining only a handful of genes compared to its free-living bacterial ancestors. This extreme reduction reflects the mitochondrion's long evolutionary history within the eukaryotic cell, a journey of integration and specialization spanning over 1 billion years. Integration into the host cell is another defining feature of endosymbiosis. Ca. Azoamicus ciliaticola, residing within the ciliate's cytoplasm, has developed a close metabolic relationship with its host. The bacterium, specialized in denitrification, generates ATP, the cellular energy currency, which it then shares with the ciliate. This energy transfer is facilitated by a specialized protein, the ATP-ADP translocase, a molecular shuttle that transports ATP across the bacterial membrane into the ciliate's cytoplasm. Interestingly, the acquisition of this translocase by Azoamicus might have involved lateral gene transfer, a process where genes are transferred horizontally between organisms, rather than vertically from parent to offspring. The potential donor in this case could be an alpha proteobacterium, perhaps a distant relative of the very bacteria that gave rise to mitochondria. Mitochondria, being fully integrated organelles, have developed even more sophisticated mechanisms for interacting with their host cells. They import the vast majority of their proteins from the host cytoplasm, relying on a complex protein import machinery that spans both the outer and inner mitochondrial membranes. This intimate integration reflects the mitochondrion's long-standing partnership with the eukaryotic cell, a relationship refined over eons of evolution. Functional specialization is a third characteristic shared by both Ca. Azoamicus ciliaticola and mitochondria. Azoamicus, having lost many biosynthetic pathways, relies on its host for essential building blocks like amino acids, nucleotides, and lipids. Its primary function, and the reason for its continued existence within the ciliate, is denitrification and ATP production. This specialization allows a zoomachus to thrive in an environment devoid of oxygen, where its nitrate-respiring capabilities become an invaluable asset. Mitochondria, similarly, have become specialized in aerobic respiration, the process of generating energy using oxygen. While they retain some remnants of their ancestral metabolic versatility, their primary role is ATP production, fueling the energy demands of the eukaryotic cell. This specialization has made mitochondria essential for the survival of most eukaryotes, highlighting the profound impact of endosymbiosis on the evolution of complex life. The potential evolutionary trajectory of Ca. Zoomachus ciliaticola raises fascinating questions. Could this bacterium, already showing signs of genome reduction and integration, eventually become a fully integrated organelle like the mitochondrion? The absence of oxygen in the ciliate's environment might have been the driving force behind the selection for this nitrate-based respiratory symbiosis. Most denitrifying organisms possess hybrid respiratory chains, capable of using both oxygen and nitrate as electron acceptors. However, both Zoomachus and its ciliate host lack terminal oxidases, enzymes essential for oxygen respiration. This suggests that they are obligately anaerobic, fully committed to a life without oxygen. This commitment, while seemingly restrictive, has opened up a new avenue for energy production, exploiting the abundant nitrate in their environment. The discovery of this symbiosis challenges our understanding of energy production in eukaryotes, demonstrating that mitochondria are not the only route to efficient respiration. It opens up the tantalizing possibility of other undiscovered prokaryotic endosymbionts, enabling eukaryotes to utilize different electron acceptors, perhaps even more exotic compounds than nitrate. This raises fundamental questions about the limits of eukaryotic adaptability and the potential diversity of energy-generating mechanisms in the microbial world. The broader implications of this discovery extend beyond the realm of microbiology. It underscores the importance of symbiosis as a driving force in evolution, shaping the diversity and adaptability of life, particularly in extreme environments. The nitrate-breathing ciliate, a testament to the power of partnership, demonstrates that even in the most challenging conditions, life finds a way, forging unlikely alliances, and constantly innovating to overcome the obstacles to survival. This discovery has opened a new chapter in the evolutionary narrative, highlighting the remarkable plasticity of eukaryotic life and the potential for even more surprising symbiotic discoveries in the future. Chapter 5. Beyond Mitochondria. Exploring the diversity of energy production in eukaryotes. This discovery opens our eyes to a hidden world of diverse metabolic strategies, particularly in environments where oxygen is scarce. Let's delve deeper into the fascinating adaptations eukaryotes have evolved to thrive in these oxygen-depleted realms. Hydrogenosomes, found in a variety of anaerobic eukaryotes, are organelles that generate energy through fermentation, a process distinct from respiration. Unlike mitochondria, which require oxygen, hydrogenosomes function in the complete absence of this life-giving gas. These intriguing organelles, often described as miniature hydrogen factories, produce hydrogen gas as a primary byproduct. But the story doesn't end there. Depending on the organism, hydrogenosomes can also generate other end products, including acetate, carbon dioxide, and formate. These organelles are not an independent evolutionary invention but are actually modified mitochondria, adapted to anaerobic conditions. They are found in diverse organisms inhabiting oxygen-free environments, such as the anaerobic ciliate Nyctotheris ovalis, which resides in the oxygen-free hindgut of cockroaches, and Plagiophylla frontata, another ciliate found in similar environments. The evolution of hydrogenosomes likely involved lateral gene transfer, where genes from other organisms, possibly bacteria, were incorporated into the mitochondrial genome, contributing to their unique metabolic capabilities. This genetic exchange has played a crucial role in shaping the diversity of energy production strategies in eukaryotes. Anaerobic respiration, another strategy employed by eukaryotes in low-oxygen environments, involves using alternative electron acceptors other than oxygen. While most organisms rely on oxygen to drive their energy production, some have evolved the remarkable ability to use other molecules, like sulfate and iron, as the final destination for electrons in their respiratory pathways. Sulfate reduction, for example, is a process where sulfate, SO42, is used as the terminal electron acceptor in respiration, ultimately producing hydrogen sulfide, H2S. This process, carried out by specialized microorganisms, occurs in environments rich in sulfate and depleted in oxygen, such as marine sediments and some freshwater habitats. These environments teem with a hidden world of organisms that thrive on this alternative form of respiration. Iron reduction, a similar strategy, involves using ferric iron, Fe3+, as the electron acceptor, producing ferrous iron, Fe2+. This process, often occurring in anaerobic soils and sediments, contributes significantly to iron cycling in these environments. Specific organisms, like certain bacteria and archaea, have evolved to specialize in iron reduction, highlighting the diversity of life's metabolic adaptations. Remarkably, even denitrification, the process employed by the nitrate-breathing ciliate and its bacterial partner, is not unique to this particular symbiosis. Foraminifera, single-celled shelled organisms found in marine environments, also utilize denitrification. Some foraminifera, like Globobulimina spp., possess an incomplete denitrification pathway, lacking some of the necessary enzymes. This incompleteness is not a hindrance but rather an opportunity for symbiotic partnerships. These foraminifera associate with bacteria that carry the missing genes, completing the denitrification pathway and enabling the foraminifera to thrive in low-oxygen marine sediments. Beyond these specialized respiratory pathways, eukaryotes have evolved other ingenious adaptations to survive in oxygen-depleted environments. Some organisms have adapted by reducing their metabolic rates, essentially slowing down their cellular machinery to conserve energy and minimize oxygen demand. Others have evolved highly efficient oxygen uptake mechanisms, maximizing their ability to extract oxygen from their surroundings, even when it's scarce. Specialized oxygen-binding proteins, like myoglobin, help store and transport oxygen within cells, ensuring a steady supply for essential metabolic processes. Research has even shown adaptations of mitochondria themselves to low-oxygen environments. For example, the anaerobic protist Pyschuia has mitochondria that have evolved to function efficiently in low-oxygen conditions, showcasing the remarkable plasticity of these organelles. The story of the nitrate-breathing ciliate and its endosymbiont, Candidatus azoomycus ciliaticola, takes on a new dimension when we consider the global distribution and diversity of its relatives. Similar gene sequences have been found in a variety of habitats around the world, from freshwater lakes and groundwater to wastewater treatment plants. This widespread distribution suggests a remarkable metabolic flexibility, an ability to adapt to different environmental conditions. Recent research has revealed that some Ca. azoomycus relatives possess the genes for both aerobic and anaerobic respiration. This metabolic versatility, the ability to switch between oxygen and nitrate respiration, provides a significant advantage, allowing these organisms to thrive in fluctuating oxygen levels. This adaptability likely contributes to their ecological success and explains their presence in such diverse environments. This dual respiratory capacity also offers intriguing insights into their evolutionary history, suggesting a possible transition from facultative anaerobic ancestors, capable of both aerobic and anaerobic respiration, to the obligately anaerobic lifestyle of Ca. azoomycus ciliaticola. The diversity of energy production strategies in eukaryotes extends far beyond the familiar oxygen-powered world of mitochondria. From hydrogenosomes humming in the guts of insects to sulfate-reducing organisms thriving in marine sediments, eukaryotes have demonstrated a remarkable ability to adapt and innovate, finding energy solutions in even the most challenging environments. These discoveries challenge our traditional understanding of eukaryotic evolution and highlight the importance of symbiosis in shaping the diversity of life. The nitrate-breathing ciliate, with its unique bacterial partner, stands as a compelling example of this evolutionary ingenuity, reminding us that the microbial world is full of surprises, and many more remarkable metabolic partnerships and adaptations likely await discovery. In oxygen-deprived lake depths, a ciliate thrives thanks to its endosymbiotic bacterium, Ca. azoomycus ciliaticola, which generates energy through denitrification, mirroring the ancient origin of mitochondria. This discovery highlights the diversity of eukaryotic energy production beyond oxygen-based respiration, showcasing adaptations like hydrogenosomes and anaerobic respiration using alternative electron acceptors. The widespread presence of Azoomycus relatives suggests metabolic flexibility and adaptability. This research expands our understanding of symbiosis, evolution, and life's resourcefulness in extreme environments, potentially inspiring new biotechnologies. What are your thoughts on this remarkable partnership? Share your questions and ideas in the comments below. Subscribe for more fascinating science stories. Never stop exploring the wonders of the natural world.